how the video works. Okay, now we finish 2/3 of the lecture. Now let's go to the last part of the lecture, gene expression. As I mentioned before, gene expression, some people simply call it protein synthesis, some people call it transcription translation, and some people call it the central dogma. So we'll talk about this. So basically what is gene expression? It's a process of using gene sequence to make protein. Now that's the definition or summary of the entire gene expression. When we are converting a gene sequence into a protein, we need different types of RNA. Remember, at the beginning of semester, when we learned about nucleic acid as one of the 4 major biological molecules, RNA is one of the examples under nucleic acid. And RNA stands for ribonucleic acid. We have 3 types of RNA that we'll cover in this class. Messenger RNA, usually we just call it mRNA. Transfer RNA, we usually just call it tRNA. Ribosomal RNA, usually we just call it rRNA. So you will see these 3 in the next few slides. DNA expression depends on 2 processes, transcription, translation. Usually this is what I give my student. We have DNA, transcribe into messenger RNA, and then messenger RNA is translate into, theoretically should be called polypeptide. Remember, protein has 4 different structural organization. Polypeptide is the primary structure of the protein. It's just a strict chain of amino acid. So DNA transcript. So this is transcription. This is translation. And transcription occurs in the nucleus. Translation occurs in the cytoplasm. And then, transcription here requires something called RNA polymerase, which is an enzyme. Okay, I will just put RNA poly. Okay, I don't have enough space to write out the entire polymerase. Okay, and then from messenger RNA to polypeptide, we need a lot of stuff. Okay, we need ribosome. We need tRNA. We need at least this. Okay, at least. We theoretically we have more than this. I'll tell you. I'll add more to this list, but let's take a look at the slides. So take transcription takes place in the nucleus. Basically, transcription is the converting DNA into a messenger RNA. Converting DNA into messenger RNA. Translation occurs in the cytoplasm. It's basically converting messenger RNA into polypeptide. So converting messenger RNA into polypeptide. And then you need tRNA, and ribosome. So let's take a look. So as you know that DNA, it is double-stranded. One strand is the coding strand, one strand is the template strand. Okay. And then DNA is converted into messenger RNA by transcription. If you look carefully, the base pair here, A matches T, G matches C, C matches G, and so on and so forth. But it didn't have this. In other words, the template strand here is transcribed into the messenger RNA. The issue is that it doesn't have A. I want to emphasize A. If the DNA has the adenine here, what will be the base pair in the messenger RNA? This is a tricky question. I've never thought about this. In the messenger RNA, it will become something called U instead of T. Remember, I gave you the complementary base pairing rule at GC, at Gentleman's Club, or at General Classroom, or at Guidance Centers. So A should be paired with T, right? You may ask. Well, this occurs only during DNA replication. When transcription occur, A pair with U. So in RNA, we do not have T. All T becomes U. U stands for uracil. So in RNA, there's no T. There's no T. We only have U. Okay. So when the DNA template is A, then after A will be transcribed into U. So remember this. And then every 3 base pair we call a codon. Why? Because each codon, for example, AGC, these 3 amino acids form a codon. AGC translate into the amino acid serine. the AGC here, I should say the AGC here. The GAC here translate into the amino acid aspartic acid. CCC here, CCC translate into the amino acid proline here. So this is the primary structure of the protein. We call it polypeptide. Well, that is in an essence the gene expression or protein synthesis or transcription translation or the central dogma. That's basically it. The rest of the video or the slides, okay, we just add the details to it. This is the backbone, big picture, the main concept that I want you to know. I want you to take home. Alright, let's look at the details of transcription, meaning that converting DNA into messenger RNA. During transcription, a gene, a segment of the DNA, serves as a template to produce an RNA molecules. Okay, they have the historical definition and proposed new definition. They are kind of complicated. I prefer the historical definition, which is easier. It's basically a gene is they call it nucleic acid. Nucleic acid can be DNA, or RNA. It's the part of the nucleic acid that can code for the protein. Basically, this is a historical definition. I like it better. I even generalize it. For me, a gene is the part of the DNA. You see, a segment of DNA that can code for protein. So I bypass the RNA molecule. I jump straight from DNA to protein. So to me, okay, the gene is a part of the DNA that codes for protein. So I bypass RNA. The historical definition basically use nucleic acid, basically telling either DNA or RNA that code for protein. The new definition, okay, genomic sequence, wow. Okay, that is the very fancy term, but basically it's like nucleic acid. It means either DNA or RNA. Yeah. Directly code for functional product, either RNA or protein. So this is relatively more scientifically accurate because we talk about, okay, the same central dogma that we mentioned, DNA become RNA, and then RNA become protein or polypeptide. So you can see that, okay, that's why, okay, they said it either RNA. Why? Because DNA goes to RNA, becomes RNA, and then RNA becomes protein. That's why the definition is like this. Yeah, so the new definition is more scientifically accurate. So messenger RNA.